All right, so this video is going to be all about nucleic acids, okay? But before we do that, let's do a little bit of... So a starch is a large carbohydrate. It is a polymer. What would be a monomer to that polymer? Glucose. Right, exactly. Glucose is a monomer. Now, whenever I ask the question, what I want you to do is pause the video, think about it, and then play the video and see if you got it right. All right, so now lipids. What are the two main parts of a lipid? The glycerol head, fatty acid tail. What about a phospholipid? Phosphate group, and then two fatty acid tails. All right, what is the word that describes the electrical charge of a phospholipid? The orientation of the electrical charges of a phospholipid. Amphipathic. The phosphate heads are positive and are hydrophilic. The fatty acid tails are hydrophobic. All right. So next, proteins. So a question for proteins would be, what is the monomer that makes up a protein? Right, an amino acid. What are the two ends of the amino acids called? On one end, you have the amine group. On the other end, you have the carboxyl group. All right. What are the types of bonds that form when you put two amino acids together? Well, they make, when you put a bunch of them together, you get a polypeptide. So it's a peptide bond that forms two amino acids together. What type of bond is a peptide bond? Is it ionic, covalent, or hydrogen? Covalent, right. It's a very strong bond. Next, what about naming the levels of the protein folding levels? Name them in order. What's the first level? Primary. All right. And then it folds into secondary. And then secondary folds into tertiary. And then this joins with another one to form quaternary, right? And remember the types of bonds that allow each one to do that. Okay, so next, now we'll get into nucleic acids. So that was just a little mini review. So nucleic acids, what are nucleic acids? Well, it's not something that you use to break stuff down with or cook with. It's not that kind of acid. It will donate a proton on one end, which is what acids do. So it technically is an acid, or they are acids. But their function is to store genetic information and to store or transfer energy. For example, DNA, RNA. These are two ways that our cells store genetic information. And then we have ATP, CAMP, NADH, and NADPH. These 4 molecules are used in energy transfer, which we'll talk more about in the next unit. All right, so two functions of nucleic acids: store genetic information and transfer of energy. The monomers of nucleic acids are called nucleotides. A nucleotide consists of a 5-carbon pentose sugar bonded to a phosphate group bonded to a nitrogenous base. So what are the 3 parts of one single nucleotide? We have the sugar, the phosphate group, and the nitrogen base. Nucleotides are monomers for what? Nucleic acids. All right, and so here's your diagram for nucleic acid. So here's our sugar. Sugar is sort of the backbone. Everything's bonded to. So, and here's our phosphate group, our PO4. And then here's our nitrogen base. So just like when we talked about steroids, when we talked about amino acids, the base structure, so this is what I mean by the base structure. For every nucleotide, this stays the same. And then this is what you change. All right, so how many different nucleotides do we have? Well, we can have an adenine here, a thymine, a cysteine, or a guanine. So where in the world did I get that from? Well, that's some prior knowledge from way back when that you should have learned. But if you did not learn that, that's okay. Now you're about to learn it. When we have DNA, remember, we have A's that go with T's, C's that go with G's. So that these will either be an A, T, a C, or G, but this part of the nucleotide will stay the same. And structure equals function, we will then, if we change this, we change the structure, and therefore we change its function. Okay? DNA and mRNA are both polymers, because there are a bunch of monomers put together, monomers being nucleotides. DNA and RNA are the primary sources of genes and heredity information. So whenever you were conceived a long time ago, you had half genetic information from one parent, half genetic information from the other parent. Those fused together to form 46 chromosomes with thousands of genes, all contained within your DNA. DNA has deoxyribose as its 5-carbon sugar. So then what do you think RNA has as its 5-carbon sugar? Ribose, not deoxyribose, but ribose. DNA is double-stranded. In eukaryotic cells, DNA is always stored inside a nuclear membrane or envelope in the nucleus. I don't know if you remember, but from the 9th grade, you have a nucleus inside of your cell with DNA inside of it. What is DNA's function? Right here, highlight this so that you know. DNA's function is to code for proteins. All right, so from DNA, we get our proteins, which is the previous thing that we just talked about. The sequence of nitrogenous bases in the DNA determines the order of the amino acids in each of the body's proteins. So don't get stressed out by too much of this stuff. Understand that a lot of the things that we're talking about now in another unit we're going to get more in depth with. Watson and Crick, two very important figures, were eventually able to determine that the DNA molecule is composed of 2 strands of nucleotide that are then twisted into a double helix. All right, the individual strands are built when the phosphate groups of the nucleotides form covalent bonds with the deoxyribose molecules of adjacent nucleotides. All right, the actual bonds form when one deoxyribose attaches to another by connecting its 5' carbon to the 3'. And this is something that we're not really going to stress about right now. The 2 strands are connected together, but is really important for the next unit. The 2 strands are connected together by hydrogen bonding with complementary nitrogen bases, and A on one strand bonds with a T on the other strand. A G on one strand bonds with a C on another strand. Each AT base pair is held together by 2 hydrogen bonds, while each GC pair is held together by 3 hydrogen bonds. All right, so just to get a visual of everything we've talked about thus far, nucleotides within strands are connected by covalent bonds. So this is on the next page, so I would scroll down. So each one of these is an individual nucleotide. Here we have a nucleotide. Here we have a nucleotide. Over here is a nucleotide. We've got the phosphate, the sugar, the nitrogenous base. All right, and each sugar of each nucleotide is bonded to the phosphate group of another nucleotide, and it creates this long chain of polymers. All right, long chain of monomers to make a polymer. And then notice here, you have your dotted lines, which represents hydrogen bonding. And so between G's and C's, you have 3 hydrogen bonds, and between A's and T's, you have 2 hydrogen bonds. So there's that we've talked about thus far. it is important to remember that hydrogen bonds are weak when compared to covalent or ionic bonds. This is important because the two DNA strands have to be separated during both processes of replication and transcription. So let's say that we are going to undergo mitosis. We're going to need for this DNA to split in half to make two new strands. So what is the importance of hydrogen bonds being here and not some other type of bond? Hydrogen bonds are weak, so they're easy to break apart to make the two new strands. If these were covalently bonded together, it would require more energy to break these apart. So you're probably wondering, if you have these two weak bonds here, how does the DNA actually stick together if they're weakly bonded? Well, remember, they're wound together in a helix structure. So just like taking two ropes or three ropes and trying to stick them together, what you do is you twist it. So then whenever you twist it, they stick together, and then you just sear the end, you smelt the ends to keep them stuck together. So that's how you make a larger rope, and that's similar to DNA. So that is how you can have these weak bonds and still have DNA to remain intact. Like a protein, a DNA molecule has directionality. So in proteins, the directionality comes from the structure of the amino acids. On one end, you had the amine group. On the other end, you had the carboxyl group. In this case, the DNA molecule is composed of two strands of nucleotides held together by hydrogen bonds. Each strand has two slightly different ends. On one end, known as a 5' end, terminates with an unbound 5' carbon on the last nucleotide chain. All right. So here would be our 5' end. So this carbon is called the 5' carbon. I don't really expect you to remember this. Later on, the expectation will be there, but right now we're just kind of getting a general idea. So DNA is structured oriented in this direction because it goes from 3' to 5'. That probably doesn't make a lot of sense. And then this side is anti-parallel. You notice the nucleotides are upside down. So here's our 3' and here's our 5'. And so this strand is oriented in that direction. All right, so that's all I'm really going to get into for that. So just for now, understand the basic structure of DNA, understand that it's made by nucleotides, get an understanding of the type of bonds that are found in between each one also. All right, and here's your DNA molecule compared to your RNA molecule. One big difference that you should notice here is that you have thymine for DNA and uracil for RNA. That's the main difference, one main difference, and another is that RNA is not a double helix. It's a single strand. Because RNA is made to be temporary inside of our cells. Not the case with viruses. You have plenty of viruses whose main source of information is RNA. But RNA, single-stranded like this, is not very stable. It's very highly mutable, which is why viruses like the coronavirus can mutate so easily, because you just have this one unstable strand versus a nice double-stranded helix. So our DNA should not be as mutable as that of viruses. All right, so the last thing, I'm not going to get into the phosphorus cycle. You guys can read that. The ATP cycle. ATP is very, very important to understand. But look at its structure. So here we have a nitrogenous base, we have a sugar, and we have 3 phosphate groups. If we take away 2 of those phosphate groups, then what do we have? Pause the video and think about it. What is this structure? It is a nucleotide. Right. So ATP is basically a nucleotide, an adenine triphosphate. It is basically a nucleotide with 3 extra phosphates. These phosphate groups are highly energetic. So ATP can be split. You can release one of those phosphate groups, release a lot of energy to initiate the chemical processes inside of the cell. So take a second to look at this diagram, and this will be the last video for this series of lectures.